Okay, so this is just a cartoon representation, right? Just trying to get this idea of the passage of information through, because that's what's important. So if you think about, you've got the DNA hidden at the back there. That's the template upon which the information that's stored that dictates how a protein folds up. So if you're driving from Cairns down to Melbourne, there's a number of different ways you can get down there. All right, and you might stop off at Coffs Harbour, and you might continue down to here. The point is, the map tells you which way you're going to go. The DNA tells you which way a protein is going to fold up. Okay, so you might have your protein sequence over here, which is dictated by the DNA. You head off down to Coffs, and you get a structure, right? And this is what we call an intermediate. And I'll come back to this a bit later on. But what I want you to get is the protein is starting to fold up. Right? As it folds up, it tries to get to those structures I showed you at the beginning. And as the journey continues, you come down to here, and this is the native state. This is the active form of that protein. This is the functional form. Now what I want you to do is contrast that with another road trip. All right? We start off with the protein up here. But if you look carefully, you can see there's some subtle differences in how it looks. I've actually changed the sequence. So I've changed the sequence of the protein. Right? This means it's going to cause a problem. And in this case, what happens is we might stop off at Rockhampton. All right? We form an intermediate. Now, if you look closely, there's differences between these two. The big difference is this thing cannot fold up properly now. All right? It's going down the wrong path because that sequence is changed such that it gets stuck where no one wants to get stuck. All right? In the middle of Canberra. And it can't go anywhere. All right? It forms these aggregates. All right? And that's because the information up here is wrong. This process does not work. All right? So what my lab is interested in for a number of proteins is trying to understand how proteins fold up. More importantly, what happens when that sequence is changed and misfolding or the incorrect folding of that protein occurs. All right? And that's the story I'm going to tell you about today for ataxin 3. My lab works on a number of diseases, and these diseases are all linked under what's now termed conformational diseases. That's because proteins have shape. If proteins change their shape, they undergo changes which lead to their sort of aggregation, the self-association, the formation of these aggregates, these fibrils. All right? And there's a whole range of diseases. All right? There's Parkinson's disease, mad cow disease, Creutzfeldt-Jakob, Alzheimer's disease, Huntington's disease, a whole range of diseases. Now, what I'm talking about today with SCAR3, you can apply to any of them. They're all very, very, very similar in the way in which the mechanisms by which these proteins change shape occurs. And one of the hopes for the field is if we can crack understanding any one of these diseases, there'll be a flow-on of information to help us with all the others. So, if we study Huntington's or we study Alzheimer's, that will have a flow-on effect to all the other diseases. So all of these diseases, you get aggregates, all right? And you can see these aggregates here. So this is protein that's not soluble. It's protein that becomes deposited in tissue, and mainly in neuronal tissue. Okay? And what's interesting is that the aggregates are what we call beta sheet based. So I said at the beginning, proteins have helices and sheets. In the aggregates, you only have sheets. All of these proteins, although we've got different proteins, all form the same types of aggregates. So here you've got aggregates of the prion protein, and they're very similar to the ones formed by ataxin 3. Okay? Subtle differences because of better microscopes and whatever else, but in essence they're the same. Different protein forms the same types of aggregates. What we also know is this thing about misfolding is intimately linked to disease, and I'll mention that as I go through. Skip over that one. Alright, so now I'll talk specifically about our working with polyglutamine diseases. Alright, so what I'm talking about here is we've got a protein which has a polyglutamine stretch. Alright, and that's represented by the Qs. So what do we mean by that? So you remember how we had the protein chain, that piece of string? On the string there were side chains pointing up and down. I said it's the sequence of those that dictates how this thing folds up. Well, one of those side chains is glutamine. Alright, we recognise it by the Q, and that's what it looks like in chemical form. When you've got polyglutamine, all it means is you've got a string of those things. Alright, so somewhere within that sequence, you have a string of glutamine residues. Now, there are about 100 proteins in the human body that have strings of glutamines. Okay? And what we know from studies, so I go back one, what we know from studies is if that polyglutamine tract is less than about 30 to 35 residues in length, everything's fine. As soon as you get above a threshold of about 37, so what that means is we have more than 37 of those glutamines in a row, that's when you get misfolding. All right, and that's when you get a range of diseases. All right, Huntington's disease, spinal muscular dystrophy, some disease that I can't say, only medics can. The spinocerebellar ataxias, which you know about. Okay, in all of those proteins, ataxin 1, 2, 3, 6, and 7, title binding protein, we have a polyglutamine tract. When that tract gets big, more than 37, and we don't really understand why it's a magic number of around 35 to 37, when that tract gets big, that's when you get disease. And that's when you also see misfolding and the formation of these aggregates, which I've already spoken about. Okay? So what we know is you get expansion of this tract, formation of these aggregates, that leads to neuronal dysfunction and leads to all the damage. Typically 30 to 50 years of age. The important thing that's important for us at a molecular level is that we know that the longer that tract, the earlier disease happens. What that means is it's more aggressive, and what it means is for us from a laboratory point of view, it's a lot easier to study because everything happens faster. All right? And so it's important for us that we know that as the tract length gets much longer, this happens faster. And I'll show you why that is as we go through this talk. All right, so what I want to get is you've got this polyglutamine tract. When it gets above 37, something happens to the protein. And that's really what we're trying to work out. What happens then, what's occurring, and how can we stop it? So what I'm going to do is tell you a couple of little stories today that we've done over the last 8 years of research. I'm only going to show you the highlights. Hopefully the highlights. So the first thing we wanted to know, and when we started this research, no one was doing the sort of stuff we did, where we were looking at the pure protein, trying to understand what's happening at the molecular level. So one of the first things we wanted to know was, could we take any old protein and put a polyglutamine tract onto it? And what would be the effect? As I said, there are 100 proteins in the body with polyglutamine tracts. We wanted to know, if you took that tract and stuck it on any old protein, does it have a similar effect to what we see in sort of the disease scenario? So we're using this as a sort of tool to try and give us clues as to what happens at the molecular level. So what we did, or Amy at the back did this work in particular for her PhD, she took a small protein, this one here, which has got 3 helices, and onto it she stuck polyglutamine tracts. All right, and she was able, within the laboratory, to produce a whole range of proteins with different length glutamines. So each one here, we've got 5 glutamines, 9, 13, 20, and all these bands represent are the different proteins she was able to produce. And obviously, as we add more glutamines, the protein gets bigger, and that's why you can see this sort of stepwise thing. All right, so all this shows you is we can produce in the laboratory these proteins, which means we can study them. Down here's the important result. So what Amy did is a very simple experiment. Is she took the protein, she put it into a test tube and just watched it. All right, she went away, had a few bottles of wine, came back, and looked at the results. All right, so what you can see here is along the bottom we have hours. All right, and on this side we have this thing called TH2. What I want to just think about that is, in essence, what we're measuring is the formation of aggregates, formation of this stuff. All right, so when you get up to 1 here, the material is 100% like this. All right, so what you can see is, if you've got 31 Qs, or we've put 31 Qs on here, there's no change in the signal. All right, as we increase the number of Qs, we go up to 35, that's this protein here. You can see there's no change for a while, and then it happens. So it's quite slow. As we increase the Qs and get past that magic threshold of around 37, you can see that the reaction occurs very, very quickly. All right? So what this tells us is, as we increase the number of Qs, this thing will aggregate and form fibrils. All right? These are the aggregates we form, and these are identical to what we form with ataxin 3. Okay? So what we were able to do with this was to replicate what we see with Huntington's, what we see with ataxin 3. The main reason we did this is it's really easy for us to produce this. It's actually very difficult to produce the real proteins. All right? But what it tells us is if you stick glutamine residues, and we stuck this in many positions within this protein. It doesn't matter where you put them. If you put them within a protein and they're longer than 35, you will replicate what we see in disease. You'll get aggregation. All right?